Thank you very much. Thank you very much for the award. It's a remarkable privilege, and I hope that I hope that it will be appropriate for a Haldane Award, because what I'm going to be talking about is not simply about science, but about how science and society have interacted, in particular over the last half century. And I also think, I can be fairly confident that Haldane would indeed be pleased that I'd won this award, because Haldane was a good Marxist, and like many other people, I'm on strike today. Okay. Now, I am going to be taking a break in the middle, because I learned that this helps enormously. So if you find yourself dozing off after about 30 minutes, there's a break coming. There's a 2-minute musical interlude in which you can listen to the music, check your phone, chat to your neighbour, do whatever you want, and then we'll start again. Okay, so what I'm going to be talking about is the way that humans have altered genes. Now, we've been doing this from the very beginning, because we're predators, we are going to eat things, and that is going to alter gene frequencies. Eventually, we mastered agriculture, and we chose certain animals and plants that we then grew and transported around the world. We were even, astonishingly, able to master, without even realizing it, biotechnology, and use microbes to make bread and beer and wine. And then finally, as we really began to understand something about how heredity works, we were able to breed, through selective breeding programs, produce animals and plants that were exactly what we wanted. So we've been doing this since the very beginning. But what I'm going to talk about is not this deep history. I'm going to talk about what happened over the last 50 years, and in particular, the beginnings and how we ended up in what I think is a genetic age, beginning in 1972, 50 years of amazing discovery and application, a revolution in our understanding and control of genes, which has had enormous repercussions on the economy, on agriculture, and on culture. It's changed the way we think about the world and we think about ourselves. I'm going to talk about the dreams, the way that we have hoped and what we've been able to do. For example, this is the very first description of being able to deliberately change the genome of change a DNA sequence in a deliberate way from 1972. And that led to, can we turn the lights down a little bit? Is that possible? That led to, I've chosen an example from my work, not my work, but my animal. This is a Drosophila. This is the brain of the fly. You can see its eyes here. And this is 2 paired nerves. And these nerves are expressing, under the control of a gene from a jellyfish, they are expressing, no, under the control of a gene from a yeast, they are expressing a gene from a jellyfish, which is why you've got this green. And what's happening here is this is basically a circuit that the fly uses to be able to decide, is it going to move, because these nerves here are controlling the 3 pairs of legs it's got. And so there's an astonishing degree of control that genetics has enabled us to exercise in fields that are far removed from genetics itself, understanding how behaviour works. Those of you who have diabetes and who use insulin, your insulin is produced through the power of genetic engineering. All insulin these days is produced in microbes, and I'll briefly be describing how that happened and what the consequences of this major development in our ability to treat people. And no matter what you might think about GMOs, the astonishing power of them. Here we've got some maize, which is expressing a natural insecticide called BT, and these are the maize plants that don't have that gene in them, and enables the maize plants to produce an insecticide and to resist the depredations of various insects. So there's been astonishing changes in the way that we've been able to do things, all to do with the power of genetics. But there's also, of course, been a downside. There have been nightmares. There have been fears. And I just want us to go through, think about 4 of them and wonder to yourself, what would you think about this? What if scientists were going to introduce DNA from a cancer-causing virus into a gut bacterium that lives in our gut? What if scientists wanted to swap DNA between any 2 species, mixing up genes without having any idea of what might happen? What if scientists wanted to make highly pathogenic viruses even more transmissible and dangerous? And what if scientists wanted to edit human embryos and then implant them? And of course, as many as you will know, all of these things happened. And what's extraordinary is that the way that science responded to these challenges, these nightmares, these potential disasters. And genetics in that respect is unique. It responded in a way that, for example, physics did not. Physicists built the atomic bomb. They got very worried about its potential deployment after the defeat of Nazi Germany, but they didn't stop working on it. And yet, in genetics, in 1971, in 1974, in 2012, and most recently in 2019, scientists have said they are not going to do experiments because they feel they're wrong or dangerous. So what I'm going to do is explore, in particular, these 4 moments where the dreams and nightmares have come clashing together and see what it is that scientists have done, and in the final part, where we're going and whether there are more nightmares out there. So the first of these events took place in 1971, when this man, Bob Pollack, who was a young postdoctoral researcher at Cold Spring Harbor, heard from one of the students on the course that he was teaching. She was a PhD student, her name was Janet Mertz, and she was the student of Paul Berg, who was a big shot professor at Stanford. And the project that Janet Mertz had been given was to, exactly as I said, to take the DNA from a virus called SV40, which causes cancer in hamster cell lines, and its effect on humans was absolutely unclear, but people were very concerned about it. And she was going to put it into E. coli. And Bob Pollack was absolutely horrified. He couldn't see what on earth would be the point of this. So he picked up this telephone, and he phoned Paul Berg on the other side of the continent and said, "Why are you doing this crazy experiment? This could have terrible effects. We have no idea." Now, Berg has got a bit of a short temper and basically told him to get lost. I think he used some fruitier language, but Pollack wouldn't tell me what the words were, and Berg said, well, I probably told him to go to hell, I can't remember. But I think basically he told him to fuck off. Berg put the phone down, had a huge row, and decided that he was not going to pay any attention to this nonsense. And then he talked to various people over the next few months. And Janet Mertz herself came back from the conference, the course that she'd been in at Cold Spring Harbor, very concerned about the potential dangers of what they were planning to do. And in the end, after about 4 or 5 months, Berg decided he wouldn't do that experiment. And he phoned up Pollack and said, okay, yeah, maybe you're right, maybe we shouldn't do that, we're not going to do it. So the first time that this was raised, this was all in private. Nobody knew about this at all. They discussed it, and they decided not to do it. Now, the reason why Berg could do that, could say, okay, I'm not going to do that experiment, is that he wasn't actually interested in that, doing that experiment. This was just a side project he'd given to a student. What he was interested in doing was published the next year, and it was the opposite experiment. He took the DNA from E. coli and put it into SV40, which could then be used to infect mammalian cells. In other words, he was trying to transform mammalian cells so he could understand how genes worked in mammals, which virtually nothing was known about. We knew a great deal about E. coli, and so he was taking a system that he knew and putting it into the mammalian cells. So to do this, he needed a vector, a way of transmitting this new DNA, and this is what this paper was about. And this is the scientific revolution, I think, of genetics. This is the moment that we stop just randomly killing animals and changing their genes. This is the moment where we begin to develop tools that enable us to precisely alter a genome in a way that we wanted to do. Within a very short amount of time, new papers emerged from both Berg's laboratory and then in particular from another Stanford laboratory and from the University of California, San Francisco, a year later, where they, in particular, Stan Cohen and Herb Boyer, used bacteria, plasmids, which are tiny little circles of DNA that you found in bacteria. They transformed the plasmids and were able to move DNA from one bacterial strain to another. And that DNA could be anything. People soon showed you could get DNA from a fly, from a frog, and you could put it into a bacterium. And this was the second moment where people said, we could do anything here. This is astonishing. Oh my God, what could go wrong? And people began to become very, very concerned. And throughout 1973 and the early part of 1974, there were a lot of discussions at conferences with people saying, we must not do this. And the eventual consequence was that Berg, along with a number of other leading scientists, US scientists, published a letter in Science and Nature, which became known as the Berg letter, because he was the first signatory, which they called for a moratorium on what was called recombinant DNA, so mixing up DNA from more than one species. A moratorium means a pause. We're not going to do this work for a certain amount of time until we are confident that it can be done safely. This led to a global debate on the possible dangers of this new form of genetic technology, including here, in this very room. So here we can see Paul Berg standing here. I'll be Paul Berg. And this was broadcast live on BBC Two at 9 o'clock in the evening. And Berg presented his ideas, why he was worried. And the audience, with an awful lot of biochemists who weren't terribly concerned about what he was suggesting, chipping in and arguing with him. And as Sidney Brenner said to him, I'm sure this isn't true of the audience tonight, I thought you carried it off extremely well considering the number of morons in the audience. That's kind of typical Sidney Brenner. So there is this generalized concern about where genetics could go. And they've organized this moratorium, which as far as we know held. Everybody just says, okay, we're not going to do those experiments. And they then have a meeting in February 1975 in order to decide how to go ahead. Really, what it was about was establishing biosecurity protocols that would enable the research to go ahead. This took place at Asilomar. This is very well known for both scientists and those of you students who were paying attention in the early pages of your genetics textbook. It's mentioned there as well, because geneticists are very, very proud of this. We've got some nice little pictures here. This is Daniel Singer, one of the lawyers who injected a note of realism into the debate. He's talking to some of the journalists who are there. This is Sidney Brenner giving a talk. And this is David Baltimore in a very fine jacket that's embroidered with roses. They don't make them like that anymore. What came out of Asilomar was firstly the use of the biohazard symbol, which had first been proposed in 1965, and secondly, a set of protocols that would enable people to carry out this research safely. And this was these 4 stages, 4 levels, from the most safe to the most dangerous, 1 to 4, had been simultaneously adopted in the UK, which had a little parliamentary committee rather than a big jamboree. They'd had a parliamentary committee that had discussed all this under the Official Secrets Act. It was most bizarre. They released their report, but the actual workings of this panel at the end of 1974, 1975 were completely secret. And this 4-stage, 4-level biosecurity protocols were summarized by Gladys Casper as don't eat it, that's the basic one, don't touch it, don't breathe it, don't do it here. And don't do it here means you've got to go to, for example, Porton Down in the UK, a germ warfare centre, to do the experiment. So the really dangerous stuff is don't do it here. Now, Asilomar is very famous because scientists love it because we self-regulated. We decided what to do. Except you think, well, wait a minute, there were 200 of you there. You decided for the whole planet. Nobody, there's no law passed. It was just you kind of arguing about it. So maybe that's not quite so good. The biosecurity measures they adopted only had really the force in America through the NIH, the National Institutes of Health, because the National Institutes of Health signed up to them and said, we will not fund any research that doesn't follow this. So, as we all know, money is what counts. So it had enormous impact on the state-funded sector in the US, but not at all on the private sector, because there was no laws. And equally, we're not bound in the UK by the NIH. But it had a great kind of moral value and was generally adopted and seen as the way forward. But just think about what wasn't discussed, and we'll see the significance of this later on. They refused to discuss anything to do with gene therapy, about manipulating humans. They refused point blank to have any discussion about bioweapons, about creating biological warfare systems that could kill people. They refused to discuss anything about potential environmental effects of the organisms that they were manipulating. So they were very clear, because they wanted to get rid of the moratorium. Many people decided it was a crazy idea in the first place. They wanted to get a way out of this mess they found themselves in, and they didn't want to discuss anything too complicated. The public were not involved. There was never a question there, is this the right thing to do? And there was no real voice of opposition. The only opposition came from people like Jim Watson, who thought the whole thing was crazy and they shouldn't be doing it in the first place. So it shouldn't be a moratorium. So there was no real deep questioning about what was going on. Nonetheless, this opened years of debate around the world, not all of which was terribly decorous. Here's an example of Godwin's law before the internet. So this is a meeting about manipulating microbes, organized by the AAAS, and protesters who are very cross about this claimed this was all about trying to create a perfect race, and you were all Hitler. So what's astonishing, as you can see, people got pretty steamed up about this all over the world. And yet, within 5 years, it had all gone. It all evaporated away. By 1980, the restrictions were lifted on all but the most dangerous experiments. You could do basically most things in category 1, I don't eat it, which is just kind of basic if you're in a lab. The safety criteria that they adopted worked well. The worries people had about putting human genes into bacteria, say, disappeared because it was discovered that most genes in eukaryotes, that's organisms with a cell nucleus, the genes are actually in bits. They're all scattered all over the chromosome, and there's lots of other stuff in between. If you take the whole thing and put it into a bacteria, the bacteria won't know what to do with it because it doesn't have the cellular machinery for stitching it all together. And above all, money had intervened, because immediately that Cohen and Boyer had shown how to do their cloning method, whereby you could put any gene into a bacterium. People had realized that there was money to be made here, and they had very, very quickly successfully commercialized first somatostatin, which is a human growth hormone, which initially turned out to have very little commercial purchase, but eventually with problems associated with Creutzfeldt-Jakob disease, because people used to get the hormone from cadavers, but when those brains were potentially infected, you wanted something pure. And so the somatostatin became a huge money spin. And above all, insulin, which, as I say, has transformed the way that this drug is delivered. And you were able to patent life forms. This was a big decision that happened in the USA in 1980, and which had ramifications throughout the world. There was a lot of money to be made. Here we can see, so Genentech was the company that was set up by Paul Berg and Bob Swanson, who was an out-of-work financier. Swanson had the idea that we can use this cloning technique. Boyer, not Berg. So he phoned up Boyer, and they set up a company the same week that Apple was incorporated in the same part of Silicon Valley. And in 1980, Genentech, before Apple, went public, and they made an absolute fortune. Herb Boyer went out and bought himself a Porsche sports car, because all of a sudden he was a multimillionaire because of the shares that they owned. And this was the biggest public sale of shares that Wall Street had ever seen. And it heralded the whole change, together with the arrival of new neoliberal policies in the governments in Britain and in America. A whole change, a sea change. It was the era of Wall Street and greed is good, prompted by Genentech. But as you can see from the San Francisco Examiner, the same day, Paul Berg won the Nobel Prize in chemistry. Berg, who's never taken a patent out for anything and done anything to make money out of his research. He won the Nobel Prize for his 1972 paper. This led to Boyer in particular, a very jovial man, becoming kind of the public face of genetic engineering, and the scientists all over the world began to lick their lips, and university administrators, of course. Because if you've done a fantastic research project, maybe you could have a startup, get a patent. So all that stuff that academics live with today, all that pressure to commercialize, has its origin in the development of genetic engineering in the second half of the 1970s. And I find this quite extraordinary. Nature, every month had a guide to bioriches, which had all the information about the latest stocks and shares and ways that you could get more money, and advising academics about what kind of balance of stocks they should have in order to make the most money. So that's the kind of origin of all this very rapid scientific discovery, precise ability to change genes. And this led to two great dreams: improving agriculture and curing genetic diseases, both of which people have been arguing about and thinking about literally for decades. The big breakthrough with agriculture came in 1983, when three groups simultaneously were able to develop recombinant plants. And one was from St. Louis, another was from Belgium, and the third one was from the big bad Monsanto. Now, Monsanto gets a very, very bad press these days, but its history and its involvement in developing plant genetic engineering is actually quite fascinating. So in the 1960s, you couldn't get more kind of American awfulness, because Monsanto, they sold AstroTurf, they sold DDT, they sold Agent Orange, which is the defoliant that was used in Vietnam. So everything that was kind of unnatural and anti-environmental was summed up in Monsanto. But in the 1970s, they decided they wanted to get out of chemicals. They recognized, and these are the terms they used in their internal documents, this is unsustainable. We cannot carry on. The world cannot carry on churning this stuff out. And that's when they had, along with the Belgians and the people in St. Louis, the idea of getting a natural insecticide, BT, I mentioned earlier on, which was used at the time. It was grown in bacteria, and organic farmers were allowed to spread it on their crops because it wasn't engineered in any way, it was organic, and it would kill caterpillars. And so they got the idea, right, we can get that BT gene, put it into a plant, and now it will be able to resist the depredations of the various insects, and we will sell less chemicals. There will be less chemicals, less insecticides distributed. So it was enormously, it was very, very sensible in terms of the future of the planet. And also they clearly thought they were going to make a lot of money. Unfortunately, they then went on to develop herbicide-resistant crops. So they developed their own herbicide and developed crops that would resist that herbicide, which, of course, rather than getting out of chemicals, involved pouring even more chemicals onto the environment. So it didn't all go particularly well. They also became extremely aggressive about prosecuting farmers. This is where a lot of their bad reputation came from. Farmers who were keeping seed, which was against the rules, against the contract you'd signed with Monsanto, because Monsanto owned the genes. You were just borrowing them. You were renting them for one generation. You couldn't keep the seeds. So they aggressively prosecuted farmers, and the whole thing got in a horrible, horrible mess. They had this bad reputation, and then in a completely unconnected moment, there was a massive food security crisis in Europe in the mid-1990s with mad cow disease and growing concerns that it might harm humans, and then the appearance of variant Creutzfeldt-Jakob disease, a horrible, horrible neurodegenerative fatal disease, which many people suspected but has still not been decisively proven, came from mad cow disease. So people immediately began to doubt and fear and be suspicious of government saying, it's all fine, you can eat it. John Gummer, the Minister for Agriculture, famously shoved a burger during the mad cow crisis into his daughter's mouth saying, eat this, and there was a photo op, and it was a complete disaster. And that kind of symbolized, don't worry about it, we know best. And there was a generalized fear and concern about what we were being told and the safety of the food we're eating. And one of the interesting things is that although we accept genetically engineered drugs like insulin or somatostatin, many, many people around the world remain unhappy about genetically modified food. It's exactly the same process, but there seems to be something different about food. There's something psychological about our relationship with food that alters how we think about it. So by the end of the 1990s, this was the kind of classic scene of Greenpeace demonstrators suited up in hazmat suits for no reason whatsoever, trashing a field of kale, GM kale, with lots and lots of police and helicopters. And of course, the tabloid press, with its usual incisive journalism, stepped in, and we had this on the cover of the Daily Mirror, because Blair said, it's going to be fine, I'd eat it, which of just, of course, made people remember John Gummer and the burger. I eat Frankenstein food, and it says the prime monster. So where are we now, 40 years later? Because it's 40 years virtually since they first developed recombinant plants. It is astonishing, the take-up of this technology by farmers all over the world, in particular in the USA. And these crops, which are primarily either killing insects through BT or are resisting herbicide, are safe to eat. That is absolutely clear. There's no evidence at all that these crops will do any harm to humans, although humans generally don't consume them, or to animals, which is mainly what they're used for. This is the success of Monsanto. I think it needs to be recognized. The use of BT crops has led to nearly 750 million kilos less insecticide being sprayed around the world. Plus the fact that you don't use the vehicles to do that, so all the carbon footprint of that distribution that would have taken place has been saved. And that is a good thing. But there's no evidence of increased productivity. It's quite bizarre. There's no evidence that we are getting more food per acre, hectare, than we were before the development of GM crops. And one of the reasons for that is the crops that they have introduced the varieties into aren't necessarily those that yield the most. They're those that are happiest to have this bit of foreign DNA sticking inside them. So there's no evidence of a massive increase in productivity. Farmers sometimes they take gambles because the insects, for example, aren't there all the time. They come and go with the cycles. So sometimes farmers don't plant BT, they plant other crops, and if they're lucky, then they save money because they haven't had to pay expensive prices. We've got, inevitably, because, evolution is smarter than we are, there are problems of resistance in both insects and plants. So there are insects that resist BT, and there are plants that resist the various proprietary herbicides. So we're actually building up a problem. And now, Monsanto, for example, knew about this, and they recommended that in order to ensure that you didn't get insect resistance, you needed to not plant every square centimetre of your field with the BT crop. You needed to leave areas where you planted the normal crop, because that way, when a mutant arose, it would mate with a mutant that could resist the BT, the insecticides. There would be other normal insects around that had bred on the non-BT crops that you were growing that could mate with, and then the gene mutation would disappear. But of course, you can only do that if you've got an absolutely massive scale of farming. You can't do that if you're a small farmer, because the type of GM, virtually all of this GM that's been done, is about resisting either insects or an herbicide that you're going to put on it. There's virtually nothing, apart from the papaya, I think. There's one GM crop that resists a disease. There are an awful lot of diseases, plant diseases out there, but that's hard. And the companies haven't gone down that route. So the type of GM, the crop strains, the methods of growth aren't appropriate for small-scale farmers. They are appropriate for the massive industrial-scale agriculture that you have in the West, in America, in France, and in the UK. And as a result, suspicion still exists around the world, including, remarkably, in China. One of the amazing things I discovered, there's a huge row going on in China. It's been going on for over a decade about GM crops, with parts of the People's Liberation Army claiming that this is an all-American plot and it's going to lead to sterility and all the rest of it, and other people saying, no, we've got to go forward and develop BT cotton in particular. But of course, they're not going to touch rice, because in China, just as in the West, there's this something about eating, in particular a staple like rice with its huge cultural significance. So we've got this situation where, one, there's been a huge take-up. It's the biggest agricultural change there's ever been, and yet it hasn't decisively altered things, and because of resistance, it's not going to last very long. What about gene therapy? Well, even before Asilomar, in 1970, somebody tried to do some gene therapy. It was unethical, it didn't work, it didn't do any harm, but it certainly didn't do any good. Same thing happened in 1980. Another rogue medic decided to chance his arm. But by the late 1980s, gene therapy protocols were first applied, enabling a handful of patients to have their lives transformed. Now, these are all done on cells that are in the body. It's not changing the whole genome of the individual. It's just on certain cells. But despite the huge excitement, by the mid-1990s, in America, NIH got very worried about all this money that it was spending to develop these protocols. And they thought there was an awful lot of hype going on. And the report said there were insufficient attention to research design, poorly defined molecular and clinical endpoints, lack of rigor. Now, I don't know about you, but if I'm going to have my genes altered, I'd really want to be certain that all those kind of points were absolutely nailed, not that people were just making it up as they went along. And then in 1999, some of you will know, a young man called Jesse Gelsinger, who had a genetic disease, but it was under control. He participated in an initial experiment, to see whether a new protocol would work. And the vector, the virus that was introducing this altered gene into his body, caused a huge reaction, and he died. And this caused an immense shockwave through the whole gene therapy community, because they'd just had their asses kicked by NIH, and now their protocols were proving to be dangerous. There were some successes. So, for example, Alan Fisher in Paris, he cured around a dozen children of SCID, S-C-I-D, which is a bubble baby disease. You can see the baby here kissing its brother. But 2 of the children developed leukemia as a consequence of where the virus had inserted its payload into the genome. Because both GM crops and gene therapy were plagued by problems due to where the vector was going to go. It could cause a mutation. That's what happened in the case of leukemia. And also the fact there was all this other gubbins floating around, marker molecules that enable the scientists to know that the procedure had worked. And this is where you get people talking about antibiotic resistance, for example, in GM crops. Because one of the markers you use to know that your plant the procedure's worked, is to use an antibiotic resistance gene, which enables you to show that the plant's okay. So there was a problem with the technology. It worked, it was astonishing, amazing development, but it was quite crude. What happened in the first 10 years, looking back, it's quite amazing. We had this technology at work, and then all of a sudden, in about 12 years, we went from this rather brutal GM approach to the gene editing with the scissors and all the business that you know about. It began in 1996 when Srinivas Chandrasekaran created what are called zinc finger enzymes. So these are enzymes which zinc fingers are the bits of the enzyme that he made. I mean, he's a very clever man, that would bind with zinc. And they would latch onto parts of the DNA sequence. So he was able to create an enzyme that would cleave DNA at a particular point, at 18 base pairs, which is pretty much, if you know an 18 base pair sequence, and it's varied, it's not AAAA, then it's going to be the only place in the genome. So this was very, very specific, and it was soon used on human cells. The precision of this led Fyodor Urnov, who was then a young man, to talk about gene editing. And it was in 2005 that this precision, compared to the shoving it in kind of approach for gene therapy and GM. Now you can actually target a particular gene and alter it and cut it in a particular way and persuade the cell to introduce some new DNA that would alter the sequence, edit it, exactly like you edit on a computer. And then in 2009, a new method, even simpler method, because the problem with zinc finger nucleases is they were hellish to program. Very, very difficult. You had to be very, very smart biochemist. And then this new technique called talons or tail ends, whatever you want to call it, that came along. It was far, far simpler. 2 papers in Science talk about cracking the code in the title and ciphers and all that kind of sexy buzzword. And this was the future. This was where it was going to go. This was what we'd all be using. And then, of course, 3 years later, without anybody looking for a new way of editing genes, CRISPR was discovered as a way, became a way of programming to edit any DNA. Became even simpler than the previous method. So very, very quickly, we had ended up with a method of very precisely altering genes, complete transformation compared to what had happened before. And this led to the CRISPR craze. This is a number of papers published on CRISPR, and this is 2012. So that's when Dowden and Charpentier published their key paper. And, well, it's kind of sloped up a little bit in 2020, I guess, because the world had other things to think about, but it's absolutely extraordinary growth. Just in case you don't know, we're very lucky that it's called CRISPR because Francisco Mojica persuaded his colleague Ruud Janssen to call it CRISPR. He said, Janssen said, yeah, CRISPR's great, much better than Spider, which they were going to call it. So we could have been spidering our genes. Instead, we're CRISPRing them. And the simplicity of this and its cheapness means that CRISPR now dominates scientific studies of gene editing. If you're going to edit a gene in the lab, then you're going to use CRISPR. That's not the case in the clinic, where you still have talon systems because it takes a long time to get a protocol that you can actually inject into a human being. So it's a bit like, the computers that they put on probes that go around Jupiter. They're out of date now, but they were very sexy at the time. So those clinical trials, many of the clinical trials that are being carried out at the moment are using old methods of gene editing, which work perfectly well. They're just not quite as sexy and precise as CRISPR. Medical treatments of CRISPR are being trialed at the moment, in particular to do with sickle cell disease. So again, this is editing genes that are in your body and in cells in your body in order to cure you of a particular genetic condition. And the first CRISPRed food is now available. It's a Japanese tomato, supposedly got extra health-giving properties. We'll see whether anything happens. But the advantage of this over the GMO is that you haven't got all the rest of the stuff floating around, the antibiotic genes and the marker genes. You have simply changed the sequence you wanted to change into something else. And with that, we take the break. So we've got genetic engineering by orchestral maneuvers in the dark. It could have been genetic engineering by X-ray specs from 12 years earlier. But there's a bit of a difference, quite interesting. X-ray specs has got polystyrene screaming about genetic engineering is going to make the perfect race. And orchestral maneuvers in the dark were genuinely optimistic. This is not an ironic song. They said, no, we thought it was great. So 2-minute break. Okay, so what I'm going to do in this last third is to talk about 3 new genetic dreams and nightmares, which have partly become possible because of this new technology we've got. And I'm going to start off talking about pathogen manipulation. Remember, these are 3 things that they refused to discuss. Literally, they refused. Well, they just didn't think about it. They said, no, we are not talking about this. And in particular, pathogen manipulation, well, it's a bit of a surprise, really. So in 1971, Alexander Baev and a man called Ovchinnikov, those of you who work on G protein-coupled receptors may have heard of him. He went on to do some great work on them. He was a biochemist. They wrote to Brezhnev. So this is before Berg's experiment. They knew exactly what was coming. And they said to Brezhnev, we need to use genetics to make new bioweapons. You need to fund genetic research in the USSR, and we will provide the military with new weapons. And in 1975, Bayev and Engelhardt, two of the people who were at Asilomar, part of the Soviet delegation, they were seen by all the bright young Americans, because these gentlemen are quite old, they're academicians. Those, literally, this is what they said to reporters. Oh, I think there's the Soviets there. Oh, those old guys, they don't understand anything. You know, it's not that they're pretending. They really don't understand. Well, I think John le Carré would have called that good cover, because these people were part of a murderous plan to use genetic engineering to create bioweapons. And they succeeded. It took a long time. They were able to, for example, they made a toxin against myelin, and they fused it with a plague bacillus, just so you could do something awful. And this effectively was going to be an infectious version of multiple sclerosis that you could then distribute, spray all over the enemy troops. They had an accident happened at Sverdlovsk. Anthrax, not genetically manipulated, but it's the basis of their studies, escaped and killed about 60 people. The Russians said, oh, it's all to do with contaminated meat. American scientists went and proved that it had come from a lab. Whether it was deliberate or not, we don't know. They were also fooling around with something that's even worse than Ebola, called Marburg virus. And one of the leading researchers gave himself a stab wound when he was manipulating Marburg. He died horribly, but his colleagues were very sensible. They extracted the virus because it had mutated while it was inside him, and they had another one for their collection. So what happened was, with the collapse of the USSR, people became very, very alarmed, very concerned that this could lead to these terrifying weapons getting in the wrong hands. Here we got the American Defense Secretary, William Cohen. You recognize the rhetoric. Regional aggressors, third-rate armies, terrorist cells, and even religious cults will wield disproportionate power by using or threaten to use biological weapons against our troops in the field or our citizens at home. And a real mania started to grip, in particular in America. In 2000, Australian researchers accidentally made mousepox vaccine resistant. Now, you probably think, well, that's to do with mice. Why do we care? Because mousepox is very, very similar to smallpox. And it was virtually certain that the same procedure would make a strain of smallpox also resistant to the vaccines we've had. After 9/11, with the enormous paranoia that gripped not only America but the whole of the West, the US massively expanded its research on pathogens. There was a huge increase in funding on studying, not in order to develop weapons, but in order to understand how these diseases might turn into pandemics. A whole generation of US researchers has been grown up in this world in which this is the thing to do. They reconstructed the 1918 flu virus. Not just they sequenced it, they reconstructed it. And terrified warnings about this is really, really easy to use. This eventually got to exactly where you'd expect. We know from documents found in Afghanistan after Al-Qaeda were beaten there that they went, oh, the West says this technology is extremely easy to use. We will try it. Now, they didn't succeed, but they wanted to. SARS outbreaks led researchers, at the beginning of the century, led researchers to focus on what are called gain-of-function studies. So trying to predict what will happen by making the virus even worse than it currently is. So we can recognize changes that are going in that direction and then intervene, say. In 2012, this is our third date, Ron Fouchier, who was a researcher in Rotterdam, got up at a meeting in Malta and said, I've done something really, really stupid. He had taken H5N1, which is bird flu, which is about 100 times more lethal than COVID, and he had now made it transmissible through the air. Now, he and a lot of his colleagues then wrote a letter to Nature that's going to sort it out, say, oh my God, we shouldn't be doing this. We must have a pause. This was scheduled for 60 days. It lasted for almost twice that time. There have been various events happened in America with leaks, inside labs, no transmission of any diseases, but a whole set of reasons started to get people very, very concerned about China and the US. And no, COVID has not been produced by genetic engineering. That is absolutely clear. But this gain of function research, which they justify in order to enable us to predict where pandemics will carry on, is still happening. Now, second nightmare, if you need another one, heritable editing. This is the fourth example, 2019. Researchers call for a moratorium on heritable genome editing. But here we've got Emmanuel Charpentier, Paul Berg, Feng Zhang, Françoise Baylis, Eric Lander, the great and the good, but there's no Jennifer Doudna, there's no David Baltimore. Not everyone agreed on this, so there is no moratorium. There was an attempt and it failed. Now, the reason for this, I'm sure you know, was that in 2018, He Jiankui, a Chinese researcher, announced the birth of 2 girls. We now know there was definitely a third one that he had edited as embryos. And these embryos, you've got to remember, were normal before he got his hands on them. He deliberately mutated them. And the procedure didn't work. The wrong mutations were introduced. They're not in all the cells. Their bodies are a mixture of kinds of cells. There were off-target effects. Other things were happening elsewhere in the genome. Nobody knows the situation of the girls. We can only hope them and their descendants, let's remember. Gene editing is forever, are okay. So how did this happen? How did we get to that situation in 2018, just 4 years ago? Well, yes, He Jiankui was a stupid, bad man. Very happy to say that. He was under enormous pressure in China for success and all the rest of it. But there's a huge responsibility elsewhere. In 2015, so just 3 years after CRISPR had been shown to work in mammalian cells, there's a big jamboree in Washington. And they talk about we need to establish a prudent path to editing the germline, not just ordinary cells, but the next generation, a prudent path. They said, yes, we need consensus. There's got to be a global agreement on this, but that's where we're going. And unlike Asilomar, they didn't define safety. I mean, whatever the problems about Asilomar, at least they said, 1, 2, 3, 4, don't eat it, and all the rest of it. None of that there. In 2017, this document came from the National Academy of Sciences. Consensus had disappeared. No definition of safety. It was still on the prudent path, to the extent that in Science magazine, this science journalist Jocelyn Kaiser described it as a yellow light for embryo editing. Yellow. Proceed with caution. Even after He Jiankui had said what he'd done, this was what they said at the outset. It was time, they said, to define a rigorous, responsible, translational pathway towards clinical trials. The whole of the community was caught up in this crazy idea that we should be editing our genes. At the same time, CRISPR was turning out to have huge problems in mammals, in particular at certain stages of the cell cycle. CRISPR doesn't work like a pair of scissors. The cell's got to respond to it in the right way. If the cell isn't in the right state, for reasons we don't understand, you can get huge, large deletions, complex rearrangements, chromosome removal in human embryos. I mean, it's absolutely terrifying. And I think the question you've got to ask yourself is, why is there all this excitement? Why at the germline? Because right now, couples with a genetic disease can have a normal child through IVF and pre-implantation screening. That's what you do. You have genetic counseling. You then make your embryo with IVF, and then you have a look at which one you want to keep. The only exceptions to this are only if both parents are homozygous for a recessive disease or one parent is homozygous for a dominant disease. Okay, so how many people are there? Well, it's very hard to know, but perhaps 100 couples around the world. That's it. That is all there is to this. There's nothing more. It's absolutely mind-boggling. All this intelligence, all this money, for Christ's sake, if you want to save lives, clean water and sewer systems all over the world, that'll do it. But it's not as sexy as changing genes. Finally, this has started to get through. This is a 2020 document from the National Academy of Science that acknowledged that the benefits will accrue to a very few prospective parents. And you know where those parents are going to be. So this is not a technology that's going to change and liberate humanity at all. In fact, as Fyodor Urnov said very tartly, human heritable genome editing is a solution in search of a problem. And yet, there's still no global agreement on a moratorium. Okay, my final point, perhaps the most scary, environmental manipulation. So things called gene drives can sweep through natural populations of things like mosquitoes that we don't like. If you wanted to create a mosquito that, for example, couldn't transmit malaria, you'd mutate it and you'd release it. It's the blue one here, and it would mate, and those genes, simply by the usual process, would simply dissipate. It doesn't work. Whereas a gene drive, which includes both DNA for where you've got to cut and DNA for the CRISPR scissors and the bit which is going to make the animal not able to transmit, the gene drive will copy itself over onto both pairs of chromosomes so that rapidly you get this exponential growth in the gene you're interested in. Quite astonishing. It works in the lab. Works in large population cases for 20 or so generations. You can crash populations, destroy them by releasing one of these things. Now, Kevin Esvelt, who was you can see, a very young PhD student, when he came up with the way of making, they'd been thought about by Austin Burt at Imperial at the beginning of the century. Kevin Esvelt was actually able to work out how to make it with CRISPR. And he said, I was pretty elated on the first day, thinking this is the answer to malaria and to so many other problems. And then the next day, I woke up and I was absolutely terrified. So I actually didn't tell anyone, not even my supervisor, for over a month. And Kevin Esvelt is now one of the main people arguing quite how dangerous this technology is and how careful we've got to be. At the same time, some other researchers who didn't know about any of these debates, they published this article in Science, and they said later on, we were thinking, when they realized what it would do, should we even publish it? And they used the right term, a chain reaction. They're trying to be clever, but it is basically an atomic bomb. Now, in 2016, there was a big report saying there's no evidence, there's no reason to release these things, but that research should continue. There's no prudent path. If the people studying human genome editing had half the intelligence and sense of these people, these entomologists and geneticists, animal geneticists, then the world might not have seen what happened in 2018. Target Malaria is trying, which is a big multinational, a lot of people from Imperial involved in this, trying to develop ways of releasing gene drives that will stop malaria being transmitted. If you want the population to know what you're doing, how can you explain what a gene drive is to a population where most people are illiterate? There is no word for gene in the local language. So, for example, it's very clever. They're going out and they're using theatre, trying to explain what it is. So you can get informed consent. But informed consent is incredibly hard to get. And there are protesters, people in Ouagadougou complaining, saying we don't want this. There's no gene drives have been released. They've simply done a control experiment of ordinary mosquitoes that had dust put on them, see whether they could recapture them, ordinary mosquitoes, after they've released them. So we're in the genetic age now. The last 50 years have transformed us, science and society. We've got cultural creations from Jurassic Park to Kazuo Ishiguro's Never Let Me Go. This is something that is deep in us. DNA is now part of our DNA, as everybody says. We can't escape it. And scientists, as I've shown, have repeatedly expressed concerns about the implications of their work. But the public, it's rather odd, especially this century. The public is now less involved than it used to be. The key issues that were not raised at Asilomar, refused to look at, have now become pressing in these three areas. Germline editing, gain-of-function studies, and gene drives, they're all much too important to be left to the scientists. The public needs to be involved. So whilst it is true that a Spider-Man, who of course was bitten by a genetically modified spider, which had been genetically modified accidentally by radiation, he said that with great power there must also come great responsibility. But scientists should not take it upon themselves like He Jiankui did, or in a way like the people at Asilomar did, to decide for the rest of us. Because this is something that everybody needs to know about and to think about. If you want to know more, there's a 3-part podcast, I think you young people call it. Actually, it's a radio program. Anyway, if you put in genetic nightmare, dreams, genetic nightmares, you can discover all about that. And then all this and much, much more is available in a book that I'm publishing in September. And that's it. Thank you.